Welcome to our first Gabather update from our own headquarters. This session will be exactly 30 minutes long. We have to leave the room after that. And I'll be the one that's presenting. It will be about 20 minutes of an update on what we're doing at Gabather, what we have learned lately. And after that, everybody can join the questions if there are any towards the end. So I'm going to move ahead with the first slide. I have actually a disclaimer here, just so in case the information that I'm giving everybody is too optimistic or too positive. It's based on the information that we have right now. And the agenda for today is actually a very short one. It's also a short meeting. I will still give a little bit of Gabather background, although I know that most of you people know Gabather very well, but I still want to set our clinical trial a little bit in a frame. And I want what everybody is waiting for, an update on the GT002 target engagement study, which we were doing now, and also a little bit on the next step moving forward based on the data that we're getting now and the status of the project. And the three musketeers that are with us today are the three usual suspects. Sorry. So I have to just lead people in as we're becoming more and more popular. I can see as the presentation goes ahead. Have to be the multitasking and well, the three usual suspects management team is myself, and then I got Christine with me and Stefan, and we are the ones that are running the Gabather. The presentation will be in English because English is the language of the presentations. Actually, all question, of course, in Swedish as you wish. And actually, English is not my mother tongue, it's actually German, but I have Stefan for Swedish and Christine for English. So we are on the safe side with everything. So enough talk. If I go ahead to the next slide, a little bit on the background. This is a little bit to share with you guys on how do we present Gabather as a company in general. So we're actually addressing mental health. I think it's a big issue. It's become a rather bigger issue in the last couple of years. We work with small molecules. That's very important as a contrast to antibodies, for example. And I think it's fair to say that we are actually quite far along on this journey with our three clinical trials, that two of them finished and one of them ongoing. And I think we got some positive results, and the beginning of them we can share with you. And the solution to addressing the mental health issues is that that we're developing compounds that affect the GABA-A receptor, and then when we're working with our lead candidate GT002, I think we have shown, as I said, preliminary results with the compound so far. Compound that we've developed is very high potency and has high selectivity, and I think it has the potential to treat a number of psychiatric disorders. We started, of course, as I show in the next slides, with the clinical phase 1 trials in our journey towards phase 2 and a potential licensing deal. And we've done since 2019, of course, Gabather has been on board much longer. Since 2019, developing two clinical trials, both the SAD study and the MAD study successfully finished. That's under the belt, and going on to the target engagement study that is ongoing, and we are about, I think, halfway through, would be fair to say. And for the follow-up compounds, of course, Gabather, we never forget, we said so much focus on GT002. We do have a whole portfolio of compounds that follow up on GT002 GT002 being the icebreaker for us and getting it through the clinical trials in order to get a partnership, some form of licensing agreement, and then revenue to put back into that pipeline that I just mentioned. So just to define ourselves where we are in the clinical phases, because that's a question that comes up more than once. It's a little bit question of semantics. Definition of phase 1 usually is that you're talking about healthy volunteers to study the general effects of a drug in a human body. And phase 2 generally is where you look at the effect of your drug that you're trying to study in patients or in the indication or the disease of interest. But you're looking at an effect. And in that case, the study I want to talk about, our target engagement study, is a little bit in between phase 1 and phase 2, because it's true that we're working with healthy volunteers, but we are looking at the effect of the drug and not only the properties of the drug in the human body. So that is, of course, relevant for this discussion. So I think it would be fair enough that we are somewhere in between phase 1 and phase 2 with a trial that is ongoing right now, in my opinion. Now, if you are, of course, a purist, you will say, no, this is phase 1 because it's healthy volunteers. I would argue, as CEO of Gabather, that we are actually in a good way into phase 2 because we're looking at drug effects in the brain with our imaging techniques. And in the phase 3 and phase 4, they're just formalities, and has been Gabather's philosophy that for the first projects, including GT002, our icebreaker, of course, will be not going into phase 3, but see that we can get a partnership during or before phase 2. So that is roughly where we are in the general picture of the clinical trials. One thing I've been, I have this in almost all the presentation, is that I cannot understand how important it is that we've been able to manage within our own R&D to make an orally available capsule for clinical trials available at a actually quite cost-effective price, which is an incredible achievement. Because if you look at all other clinical trials where you have to give the compound either in other formulations or over longer time, if you go and approach any clinic and say, wow, do you have a clinical trial material that is orally available? That's fantastic. That's the first good news. Second good news, of course, that what we have learned from the clinical trials so far and what we're learning with the clinical trial that is ongoing, it's actually well tolerated. We have not seen any adverse effects so far in anybody, which is very nice. So the drug is safe and it is able to upscale. Seems to be a trivial processing. It's actually not. So it's actually very good that has been going across. And we have developed our first product forms, and actually we've made it part of our patent portfolio just to show how important that is. It wouldn't have been the first drug candidate that actually does not succeed because of a problem with formulation. So that is very good. We got the clinical trial material, we got the clinical trials, and now we are going into looking into the effectiveness of GT002 in healthy volunteers. And we've made a decision at Gabather actually quite a while ago that what we are going to do, we're going to use EEG, i.e. electroencephalogram, the recording of the electricity in the brain, as our biomarker. And biomarker just means is what does the drug do to the body? It's a marker for this efficacy I was talking about, and for selecting our drug for mental disorders. Mental disorders is, of course, non sequitur that we're looking at the brain. So looking at the electrical activity of the brain is the key thing for us to look at the effect of the drug. We started with more looking at the activity in healthy volunteers. The pattern that we can see there induced by our candidate drug, we then want to transfer into activity in patients in clinical trials further on as the next step, looking forward at GT002 effects in the clinic. And in EEG, just very short, and I just want to point out this cap that patients or volunteers need to do when you do an EEG recording. It's not a trivial matter to actually just adjust this cap. You can see there are a lot of electrodes that you have to attach to the skull. It needs quite some expertise actually and technical skill to do this properly to get good data in, and the data is then different frequencies of electrical activity in the brain and in different areas in the brain. So we do EEG because it's a very fast way of recording electrical activity in the brain. So there we get the temporal aspect of the modulation of the brain in disease, and hopefully the modulation with our drug. And this is how it looks in a more in a picture. If you do look at the activity, red shows high activity and blue shows low activity. And then you can see what you can do. And this is a generic picture. It's nothing to do with GT002, but it shows you the principle of what we're doing is that you look at the activity in the brain in a normal situation in different frequencies, delta, theta, alpha, and so on. And then you compare this activity at that frequency with a mental disorder, and you can see the differences between the normal and the disease state. And it's these differences that we will try to address, to correct or to modulate with GT002. That is our philosophy and our approach that we have chosen compared to other biomarkers that you can use, i.e. you can use questionnaires, you can use tasks that people have to do. But we believe that this is the window into the brain, and a window both into the healthy and into the diseased brain. And it will also be a window and indicator of the therapeutic effect of our drugs. That is our hope, that we decided for that some years ago, and we've chosen a partner, a CRO, which I'm going to talk about now, to run this kind of studies for us, because it's nothing that we can do in-house, in contrast to the preclinical trials. And this is an example of what we want to do. It's called a lock and key approach. What we want to do is we want to change the pattern that we can see in a disease state by treating with our drug into a pattern that you can have in a healthy state. And this is what you can see. It has to match the pattern in a healthy state induced by our drug, change it from a disease to a healthy pattern. That is what we want to do with GT002. We have put in GTX here because it's a generic effect that we hope to see with our compounds. And of course, each disease has a specific pathology, a pathological pattern, and that is that we want to address with EEG. That's why the EEG is a very general biomarker. It will be a very general way of showing what is wrong in the EEG signal in a disease, and how can we address this with GT002. And all this in human and healthy volunteers and in patients, of course. And this is the example of our where we want to get at, and this is a little bit of future music, and we're on the way there. Again, this is an EEG pattern. You have different frequencies, and you compare a normal with a mental disorder state. You can see the differences. Here, for example, very low activity, you have very high activity here. And what we hope to get is that once we treat with GT002, for example, or any of our other compounds, then we will compare again. And what we want to achieve, of course, is that with the treatment with our drug, you restore the pattern that you have in a normal brain, like, for example, this one, back with GT002, back to a normal as compared from a diseased brain. Or, and this will not be perfect, of course, but take this example. Here we have a healthy alpha frequency band. Here you have a diseased alpha frequency band in an EEG. And if you GT002, we hope that we can restore, make this pattern look back to the healthy or the normal pattern. That is the whole idea behind our emphasis on the imaging with GT002, and this is basically the same in a numerical way, and this is the image of the same data that we expect to get. So for the ongoing status of for the GT002 study, we've done a due diligence visit, which I want to talk a little bit about. We went to visit our partners at the 2CA CO in Braga. Maybe it's a background, actually interesting enough, that Braga is an academia private collaboration. So it's part of the University of Minho in Portugal. There's a very competent group, and if you look them up, you would see that they are very, very active in a large number of European consortia. So they're very much ahead and present. You can check it up on LinkedIn if you wanted to in imaging studies. in Europe. So I think it was a very good partner that we found there. We spent quite a long time there. The whole team was in Braga. We reviewed the process that they have. We had a lot of meetings with the personnel. We checked out the project management. We checked out the patient journals or the healthy volunteer journals. How do they do the data acquisition, and how do the analytical process? Because the analysis of imaging data is actually very complex, almost a little science by itself. And the collected data that we have so far, just as a reminder, this is actually a blinded study, so we can only get so far with what we can predict by the analysis of the data. The results are good. The quality of the data is good. And regarding the modulation of networks, I think the results come to be very interesting with the guesstimates that we're doing with the data that's coming in. The review data so far that we have is not large enough and not conclusive enough because it's not unblinded to draw final results, but preliminary results look, quality of data is good. And at least in some of the neural networks, it seems to be a very, very interesting indication. Interesting enough, actually, that we want to move ahead and use this imaging data that we have already now for example, one application and other collaborations. And now we're talking about patient studies in the future. The study timeline, we all know that this has taken longer time that we wanted, and with a lot of discussion that we had with the group there on how we can do this, how we can improve this. I talk about this more on our next slide. And one thing that we learned actually when we were down there, that EEG and fMRI study, never mind the intricacies of getting them, they are still, in my opinion, and that was actually confirmed by what we saw down there, is that it's the best data you can get, in my opinion, regarding efficacy of a drug in the CNS if you compare it with a questionnaire or an interview or a blood sample that you could or could not do regarding CNS disorder. So I think it's absolutely brilliant. And the concept of a window into the mind, a window into the brain, is definitely true. And on the lower right-hand side, I know it's very small, at least on my screen. All I did here was just to show you that the process, and that is what I'm going to go on the next slide, of acquiring the data of both EEG and fMRI, i.e. EEG and magnetic resonance imaging at the same time in a healthy volunteer, is much more intricate than you would think of just doing, oh, it's just a question of putting a person into this tube and clicking on a button. It takes the whole process. You need one person to be there for at least between 4 and 6 hours. And the trick in this thing is that the whole process from starting to putting on the EEG cap that I showed at the beginning to getting the fMRI data out has to be perfect in all situations. So the volunteer has to come in time. The EEG recording has to be good. It has to be in the fMRI tube without moving. It has to be able. They have to be answered the question and tasks we give them. So it's an enormous process, and the quality of the data depends that this entire process works like a clockwork from A to Z. And of course, that is not really realistic in real life, and that's why it's so difficult. And actually, it takes time to acquire a good, proper data set from one person alone. And if you think that in our study, it has to be the same person that comes in 3 times with a couple of weeks in between the measurements in order to make it their own internal control, the complexity of getting one good data set out is very large and very tricky. And I think that has been a little bit of a challenge for us, because you can imagine that you get a person starts a trial, does 2 recordings and fails to show up for the 3rd one, then you're back in scratch and on step 1, because you have to start with a new person. The data is not complete. So the completion and the quality of the data, they have been the big challenge for us so far. We are getting there, and I'm still convinced that this is the right way to do it, because the data that you get out in the end, once you have gone through all these loops and hoops, is so extremely good and so much more relevant than any other data that you could get in a very quick and easier way. That is always the way I would choose it. And in the last slide, I think this is very relevant. for the partnering process. At the end of the day, we want to partner this project eventually, and it will be based on the clinical trials. So the team that we met, I think they're very competent. They're very engaged also in this study. And this picture on the right-hand side is actually a real person during our visit doing a real recording of MRI in an MRI tube. I can give you an example of the challenge. If a person in an fMRI tube is not completely quiet, silent, and still, the data becomes very difficult to interpret, and the amount of space that person is able to move in a tube is very little. We're talking millimeters here. So if you have a person, for example, that's very nervous to be pushed into a tube, and they move too much, you're back to scratch because you can't use the data. Just as an example of the challenges you have to get proper data out. Once you have the data, then you're on top of things, of course. We agreed with our partners to make a new data acquisition schedule, so we are prioritized. This equipment is, of course, in very high demand because everybody wants to use the combination of EEG and fMRI in their study for the next couple of months during the summer. The team here now, together with them, we've acquired the know-how and actually started to transfer the data from the site in Portugal to our own lab and our own servers here, so we can start the data analysis and accelerate that with working on it ourselves. However, this process is time-consuming, and it includes the whole team, i.e., it's both Stefan and myself and Christine that are working on this. The data sets are enormously big. You can imagine if you combine both EEG, so we're talking gigabyte and gigabyte and gigabyte. And we, of course, at the same time, learning also for us, we're trying to see how we can use development in AI for speeding up the data processing. This is going on quite rapidly, as everybody knows. We have an eye on this, but right now we're focusing on supporting the data analysis so far. And again, in the lower bottom here, this is a generic picture of the workflow of an MRI, fMRI process. And you can see from the experimental design to the very last step in the data analysis is actually a long way. Again, I know I'm repeating myself, but again, all of these steps have to be okay in order to get a good data set out at the very end. And that is the one take-home message that, okay, we all knew this from the beginning. If you have bad data into the analysis, you can only get bad data out. So good data in, good data out. I think it's very, very important. And if I go ahead with the next step, what are our next steps? We have a annual grand meeting of the shareholders coming up on next Friday. The nomination committee has reinforced our board for the next step, which are, of course, agreements and licensing agreements with bigger partners. So we have two candidates as new board members that will help us both with the legal aspect, administrative aspect, regulatory aspects, because the team that we have here, we all have a scientific background, so we can now, of course, push the science. But I think it's about time with the data that's coming out, and I think it's a good time right now to also then reinforce the legal aspects, the commercial aspects for the out-licensing of the project. And the idea, if you think about the next steps towards the future, is that we will use the data for the GT002 and the patterns that I've been talking about on the EEG and the fMRI for the other indications that are possible. I will see how does GT002 modulate the patterns of the neural circuits in the brain. And once we know that, we know which indications are relevant for this kind of patterns where you do have deficits. And then we will use the key and lock philosophy that I talked about earlier, and we will then see if we can restore the patterns to the normal patterns or the healthy patterns with GT002 and the other drugs. So GT002 will be interesting for a whole variety of indications. We started at the very beginning of Gabather with looking at things like schizophrenia. We're looking at things like dementia, Alzheimer's disease. All these diseases have changes in the patterns. in the brain between the different neural networks. And these are the ones that we will address with GT002. And GT002 will then be sort of the reference compound for the follow-up compounds, where we'll do the same thing, the same approach, the same philosophy, by using imaging as a biomarker to get into the clinical phase 2 patients. And the data that we have got already now has at least enabled us to looking for, or being actually invited, if I put for myself correctly, to look for soft money applications in larger consortia to look at the effect of GT002 in the first case, in cases of dementia, different forms of them, which might be very relevant, and that will be then funded clinical trials in phase 2 in patients based on the imaging data and using, again, imaging approach as a biomarker. And we would, for example, look at the pattern in a neural network in a demented brain. In the case of a dementia syndrome, then we would look what is the effect of the GT002 in restoring the patterns based on the data from the target engagement study. And this is, of course, the target engagement study that is so relevant for the potential partners in the future. So I think I'm at the very end of this little bit tour de force presentation. Again, this is as a last slide. Our idea, EEG and fMRI are in the center of our biomarker activity. The changes that you can modulate and you can induce with our drugs in this EEG and fMRI can be relevant for this whole range of diseases that you have around. It can be anything from schizophrenia, dementia, depression, and so on. It's always the same approach, restoring the normal function of the brain by looking at biomarker imaging approach. And I think GT002 is very good on the way with that. And hopefully, then once we got the GT002 data, we can do it with the other compounds that we have coming out. And finally, this is a lot of text. The key story here is, as I said before, that the success of reaching partnerships depends on the success of the results of the clinical trials. And the success of the clinical trials depend on the quality of the data that we're getting out. And I think we're getting the best quality data, and that, of course, takes longer time than we had hoped and expected. And I think that the successful partnerships, of course, they are the cornerstones, or the stepping stones, at least, in our journey. And I think that's the very last. I know that I left almost no time for anything, but I thank you for your attention. And